Greetings, teacher. I am Anjali Athar, pursuing Basic Zoology Honours from Maitri College. My roll number is 0140, and today I am going to deal with the topic Neuromuscular Junction. In this video, I would be covering definition, structure of neuromuscular junction, acetylcholine, receptors, ion channels, and most important neuromuscular transmission and some diseases. A neuromuscular junction is a chemical synapse which is between a somatic motor neuron and a skeletal muscle fiber, and it is also known as myoneural junction. Now let's have a look at its structure. There is a terminal end of the neuron and the surface of the muscle membrane with a space between them where they can communicate. It's a synapse, so the space between the two forms a synaptic cleft. The neuronal membrane before the synapse forms the presynaptic membrane, and the muscle fiber membrane after the space forms the postsynaptic membrane. The neuromuscular junction is a chemical synapse. Why? Because it uses a chemical neurotransmitter for communication, which is acetylcholine. It can get synthesized in the synaptic terminal itself. Acetylcholine is a small molecule neurotransmitter. It gets synthesized in the terminal itself and then packaged into vesicles. But the vesicles with the proteins in their walls, those are formed in the cell body and then transported down. Acetylcholine is synthesized by acetyl coenzyme A plus choline, and the enzyme that works here is choline acetyl transferase. ATP is hydrolyzed to bring the hydrogen ions into the lumen against the concentration gradient. Then, by an acetylcholine hydrogen ion exchanger, the hydrogen ions leave the vesicle and the acetylcholine enters. These are called synaptic vesicle, and each synaptic vesicle has got lots of acetylcholine molecules in it. Since energy is needed for the synthesis of acetylcholine, there is also mitochondria in the synaptic terminal. These vesicles are going to fuse with the membrane in order to release their content. The sites at which they fuse are called the active zones. What makes the fusion happen is calcium. So on the membrane, there are also voltage-gated calcium channels. This whole thing forms our presynaptic terminal. The postsynaptic membrane is formed by the muscle fiber membrane. Now the portion of the membrane that forms the synapse is thickened and it's thrown into folds. These are called junctional folds. The folds increase the surface area. At the crest of the folds, there are receptors for acetylcholine. These are ligand-gated channels, which means they open when a ligand binds. The ligand here is acetylcholine. Two molecules of acetylcholine have to bind for this channel to open. This is a cation channel, so it only lets positive ions pass through. The important ones would be sodium and potassium. The resting membrane potential of a skeletal muscle is negative, so the driving force on the ions moves sodium into the cell and potassium out, but it's mostly sodium entering. Let's understand the neuromuscular transmission now. When an action potential arrives at the synaptic terminal, there is depolarization from sodium entering the cell. That potential change opens the voltage-gated calcium channels, which let calcium ions enter the cell. The entry of calcium ions makes the synaptic vesicle filled with neurotransmitter to move towards the membrane. So the vesicles now move towards and fuse with the presynaptic membrane to discharge acetylcholine into the synaptic cleft. This process is exocytosis. Now acetylcholine binds to its receptors when two molecules bind. The ligand-gated channel opens and sodium enters the cell and potassium leaves. But it's mainly sodium entering that we need here because that is causing the depolarization. The potential becomes more positive. This is an end-plate potential, which is an excitatory potential. Depending on the calcium influx, the vesicles release the neurotransmitters and lots of vesicles attach to the membrane. So the potential sum it causes creates an end-plate potential. This potential leads to local current spreading, and when the membrane potential reaches threshold, that opens the voltage-gated sodium channels, causing the sodium ions to rush into the cell. There is depolarization and an action potential on the muscle membrane. Now this action potential is going to get propagated along the muscle membrane, eventually causing contraction of the muscle. That process is called excitation-contraction coupling. But what happens to the acetylcholine? There is an enzyme in the synaptic cleft called acetylcholine esterase. This enzyme breaks down acetylcholine into acetate and choline. The choline then gets taken up by the presynaptic terminal, that's by a sodium choline cotransporter, so it can be used to form more acetylcholine to be released when the next action potential arises to start the process all over again. Now let's talk about diseases related to neuromuscular junction. The first and most important one is myasthenia gravis. The other diseases are Lambert-Eaton myasthenic syndrome, neuromyotonia, congenital myasthenia, and botulism. Here are the references, and thank you for watching.